it's voltage gated or if it's ligand gated. It makes a difference, right? So anyway, that calcium is going to come in, trigger the synaptic vesicles that contain neurotransmitter to release the neurotransmitter using exocytosis into that synaptic cleft. And then that neurotransmitter is our ligand. And now we switch over. We're going to pick, right, something different from the pie. And we're going to pick some sort of ligand-gated ion channel to embed in this postsynaptic membrane. So the ligand binds, right? You see that there, and that will trigger the shape change. Usually the channel's going to open, right? And whatever ion that is built for, might be sodium, might be chloride, right? We'll see different things in different locations. That will enter in and create our graded potential. Graded potential over here along the inside surface of this membrane. Good. So the thing to remember is, or one of the things to remember is action potential, graded potential. And then we can sum all of our graded potentials. We can generate many, many, many graded potentials. We can add them up, right? Sum them, summation. And then all of those are going to travel to, let's pretend we have an axon hillock down here, right? We've got this whole cell body of the neuron, and we've got an axon hillock at the bottom. So at the axon hillock is where we're going to sum all of the graded potentials. Whatever it adds up to is whatever it adds up to, right? If at the axon hillock it adds up to some sort of voltage that will bring that section of membrane to minus 55, right? Minus 55. Remember that was our threshold. And once we hit minus 55, we're going to go. We're going to commit to an action potential. So we can create the action potential down here if we hit minus 55. If not, like if we only hit minus 45, oh no, not minus 45, minus, let's make it 65, right? Minus 65, too bad, right? Minus 60, no. But 55, good. Action potential. So from there, we would create another action potential, propagate it, right? Get it to move down the axon, and then it's going to show up at the next synapse, right? That would be like on the first floor, right? Synapse on the first floor, and we'll have this same sequence repeat itself, right? Action potential arrives, trigger calcium, trigger neurotransmitter release, and then we've got our ligand-gated channels. We create our next set of graded potentials. We can add those up, and you can keep going. You can have this chain, right, of neurons, and at each synapse you have this happening. Good? All right. So, all right, and we reviewed, right, or we learned about EPSPs, IPSPs, and there's adding, and there were those videos with the animation, so you can take a look at those over, oh my goodness, the weekend. And then we had ways of controlling the synapse, and we talked about like this, right? We didn't do this? Oh, really? Oh my gosh, where did we end up?
At the picture.
Oh, that's where we ended up. Oh my gosh, okay, wait, whoa, let's backtrack. Oh my goodness. I could have, oh, that's right. Let's go back. We'll talk about it. Hang on. So we ended at, did we at least do the summing?
No.
We didn't do the summing?
No.
Oh my goodness. Oh, jeez, you guys. Oh, I'm so sorry. Let's see.
We're, yeah, 31. We ended up at 31.
We ended at 31. Oh, why did I think we at least got to that?
We did this one and then the next one.
Did we do this one?
Yeah.
Okay, we did this one.
And that was it.
Did we didn't do summing? Ah, okay. So sorry. Okay, let's do summing. So we saw before that we had, okay.
Quick question before we're going. Can we also get that action potential happens at presynaptic and then graded potential happens at postsynaptic?
I think you could say that. I think you could
Like
sort
the
of
dendrite
make
in
that
the body?
statement. Huh?
Like the dendrite in the body?
Yeah, so the dendrite in the body, right, those are going to be postsynaptic,
Okay.
right? Yeah. Good. Okay, so what do we got here? Summing. All right, so we already know EPSPs, right? The depolarizing shape of a graded potential. And IPSPs were the hyperpolarizing shape, right, of a graded potential. So now let's take a look at different ways that we can add different shapes together of these graded potentials. Here they're giving us a neuron that is receiving input from 3 different axon terminals, and they're just giving us A, B, and C. So A and B also colored in green to represent excitatory, right? EPSPs. Do not talk while I'm talking, ladies. They're also relatively close to each other, right? So we know that. And then we've got C represented in red, which means inhibitory, right? Inhibitory. And I don't know, that seems to be on the other side, right, of that neuron. So let's see all sorts of different things that we can do and how we can add these things together. So we've got, if we release some neurotransmitter from A, right, from A, at this point in time, we might get an EPSP. Let's say it looks like that, right? And then we let a little bit of time go by, and then we release sort of a second batch of neurotransmitter. And then we would get, oh, it looks like we have another EPSP. I suppose it's slightly different, you know, maybe it's a little bit bigger, right? But it's not too much bigger, right? So now let's have a different situation, and let's release neurotransmitter from A, and then very soon after, let's do that again, right? Let's release neurotransmitter from A again. And now we see here's our first EPSP, but that release of the second batch happened before that EPSP was sort of finished, right? So in that case, when that happens, you can right away add stuff together so that, oh my goodness, now you have this really big thing. You have this really big EPSP that you added together because from the same axon terminal, we release 2 batches of neurotransmitter, like boom, boom, one right after another. And we can add that together and then get a big EPSP. All right, so that's one way that we can sum graded potentials by putting them close together in time. All right, let's look over here. Now we're going to release from B, right? And we have that EPSP. All right, fine. And now here we're going to release from A and B, both of them together at the same time, right? And because they are relatively close together, right? They're on these patches of cell membrane that are kind of continuous, right? So, we see, again, adding together the graded potential from A and the graded potential from B, and now we have one big one, right? Because they were close enough together that they affected each other, right? And then you ended up with one big one. So that's what we're seeing here. So that's another way of adding graded potentials together when they're close enough to affect each other physically, like close enough in space. All right, so we got that. And now let's look over here. Let's kind of do all that stuff all together, and we'll have AA and BB, right, all squished together. And it looks like, wow, we're adding, and we hit threshold. And there's our action potential. You see that? So if this situation happens, like I just said, at that axon hillock, right, we add it up, and it's enough to hit minus 55. Let's create an action potential.
So, but relative refractory period doesn't allow you to have another action potential while one is going on.
Right.
Like these can add up to create an action potential.
Exactly, exactly. So we've got 2 very separate things happening, right? Here we've got graded potentials. They're all being generated all along the cell membrane in the cell body and dendrites, right? And dendrites. Over here, they're all going to travel to that axon hillock, and that's really where you're going to do all of your adding, right? And if it adds up to hit minus 55 at the axon hillock, we can go ahead and create an action potential. We're not going to create action potentials along the cell body because of the setup. We're not going to put, that pairing of the sodium channels and the potassium channels, right, the voltage gated ones. We're not going to even put them over here, but we're going to super concentrate them at the axon hillock and along the axon. So there we have the equipment to create an action potential, right? All over here along the cell body and the dendrites, we're just going to put in the equipment, right, the ion channels that are capable of creating a graded potential, the ligand gate, right, ones. Good. So depending on what you pick from the pie, that determines what you can do, right, what you can create. Can you even create an action potential if you don't have any voltage-gated sodium ion channels and voltage-gated potassium channels? Like, can you even create one if you don't have them there? No, right? So you have to sort of straighten out, right? We have to for sure straighten out in our heads. Okay, what do we have here, and what do we have over here, right? So here, how do we build? So here in the cell body and the dendrites, we're really just focusing on the ligand-gated ion channels, right? So that we have neurotransmitter, our ligand, binding, and then we open up the channel, and then some ion, right? Different locations will have different ones. Some ion will enter and change that voltage on the inside of the membrane and create your graded potential, right? Could be this, depending on what it is that just came in, right? So, we've got that situation here because that's what we chose from our pie. Right? We chose those channels from our pie. What we did not choose in this location, we did not choose that pairing of voltage-gated sodium and voltage-gated potassium channels. That pairing is what we need for action potentials, but if we don't put them in a location, we can't make an action potential, right? So we're not gonna, right? So don't put them there. Let's not put them there. But we are going to put them in the axon hillock and along the axon, right? That's where we want to create the action potential, because here we're just making a decision of like, whoa, what is all this information coming in, right? We're getting all this here. They're showing us information coming in from 3 directions, right? Let's say that's 3 different neurons telling us stuff, right, from the outside. Good? And that relays the information. You're using neurotransmitters, which is the rest of the day. That's the rest of the day's topic. And we create these graded potentials, right? So that's our information that we take in, the graded potentials. And we add them all up, figure out, right? Send all those signals to axon hillock. Axon hillock adds it up, and depending on the sum, the number, decides, should I send that information or should I not send that information, right? And the decision is based on hitting minus 55, right? Hitting threshold. If we hit threshold, I'll send this information, right? We don't hit threshold. Oh, well. I just took in all that information. I just did all that math for nothing, right? Maybe it's not for nothing. Maybe it's not important to the body, that whatever we took in, right? And that's okay if something is not a strong enough stimulus to send that up to the brain and distract, right? Distract the brain from that piece of information. Maybe it's not that important, right? That's kind of good. Like if you're trying to focus on taking this exam, right? Then, yeah, you don't want every single last thing to be coming in at you, right? And registering in your consciousness, right? You don't want all the distractions coming in. You just only want the exam, right? Coming in. Good? So it's kind of that idea. So if this is where we're going to make our decision, like, are we going to gonna hit threshold and create an action potential or not, right? What do we have to put in the membrane of that location if we're thinking, sometimes we're gonna create an action potential, right? We're gonna pick that pairing of the voltage-gated sodium and voltage-gated potassium ion channels. So you build them in, and then you've got your capability, right, to create an action potential. Good? All right, go ahead.
Are there some in categories of significance? The temporary.
Definitely,
Okay.
definitely. Like we'll see, especially it'll show up more next week
Okay.
when we talk about sensory stuff. This is going to come back for sure and haunt us. So definitely know, temporal is time, where you put the stimuli close together in time,
Okay.
right? And then they would add up to something bigger, right? And also spatial is like physically, location-wise, close together, and they can affect each other and add up. Good? All right, but yeah, this will come back to haunt us next week. So, let's see. So far we got to this point where we had AA, right, close together in time, and also BB, close together in time, but A and B we also know are close together physically in space. So we, I don't know, happen to do something like that. We can add. Oh, and this one happens to bring us to the minus 55, our threshold, right, millivolts, and boom, right, at that axon hillock where we're measuring, right, that's our little electrode from the voltmeter measuring over there. So, boom, we create our action potential using our voltage-gated sodium and voltage-gated potassium ion channels, right? And we make our action potential, and of course we can continue, right, down the axon. See, we can continue, of course it would continue down the axon. Once you create that action potential, we already know that an action potential is all or nothing. So if we're gonna start to generate an action potential, we're gonna finish it. And if we generate one action potential, okay, let's say axon hillock, then we can send that down, right? And we will send that down to the next segment, right, which we saw, the next segment. And then if we have myelin, we can make it go even faster. Good? All right, so let's look over here, and we see from C, right, our inhibitory synapse, which is releasing some neurotransmitter that ends up having the effect of hyperpolarization, so we get more of the valley shape instead of the hill, right, shape. So if we have an inhibitory synapse creating an inhibitory postsynaptic potential, there it is, right, our downward blip over there. If we were to combine, they just happen to pick, let's do A and C together. I mean, they could have also done B and C together. They could have done A, B, and C together, right, but they're just showing A and C, fine. We know what A can create on its own, and now we know what C can create on its own. If they're like pretty close to each other, right? Positive and negative, but pretty much the same size, then here in this example, they cancel each other out, right? It's like adding +3 plus -3 equals 0, right? So that's just what they're showing us here. Good? So whatever it is, and you just add it up. It's just arithmetic.
So the inhibiting synapses in C on this graph isn't necessarily happening from an overcompensation. Like when you're saying AT, the sodium, potassium, ATPase pump would have to like work harder to compensate for potassium being sluggish, and because it would dip down and go down.
Yeah, so that has nothing to do with
Okay.
with graded
Okay.
potentials, right? That those sluggish
Mhm.
potassium ion channels, those are the voltage-gated ones that work to create an action
Okay.
potential. And once we get to this hyperpolarization here that is part of an action potential, yeah, here sodium potassium pump, ATPase pump, get back to normal so we can create another
Okay.
action. So we're ready for another action potential. But here, an IPSP is a completely different entity, right? That we generated in a completely different way. Good. So definitely, these are really good questions because you want to be sure, right? Not just for Tuesday, but also for the rest of the course. Because nervous system is going to, rear its ugly head, every other system. So you definitely want to have this stuff. This is our super basic stuff. Everything from here on in is dependent upon, is dependent upon the pie, right? And this is our first time of using the pie, right? Our first system where we're using the pie. So you definitely want to be clear on if we pick this, we can do this. If we pick this, we can do this, but maybe not that.
So the ATP pump is action potential, and then IPSP is graded.
So the
Graded
sodium potassium ATPase pump,
Yeah.
right? What was that? Let's review because that's the basis of
That was
everything.
primary.
That was primary active transport, and what were we setting up with that? Like, what did we use that sodium potassium ATPase pump to set up in every cell of the body, not just nerve cells, not just neurons? Membrane potential, maybe just one more word in there. I mean, membrane
Active?
potential is correct. Huh? Maintenance, right? Let's kind of stick with membrane potential, right? Membrane potential, that was that positive charge clinging to the outside and the negative charge clinging to the inside, right? So for every cell, right, in the body, we'll have sodium potassium ATPase pumps generating that, right, positive and negative clinging charges on either side of the membrane. And we call that membrane potential. When we look specifically at neurons, we add on our additional word resting, membrane potential. And we say that, most neurons are going to hang out for their resting when they're not generating an action potential. Like, maybe think of it this way. Action is doing, resting is not, right? Not doing. It's resting. It's not creating an action potential. So when a neuron is not creating an action potential, right? It's resting, right? It would be resting. So, we have said that for kind of, when you look at all neurons, right, sort of, we don't pick any particular one. That resting membrane potential could be maybe at minus 90, or it could be maybe at minus 40, right? We had a range. But we are going to focus on kind of most of the neurons, right? Most of the neurons are actually going to hang out at minus 70. So that's what we've been looking at as our most common resting membrane potential for neurons, right? Is that minus 70 millivolts. We'll see next week. I think we'll look at like one exception to that. I hate to say rule, you know what I mean? But to that sort of most common situation, right? I think we'll look at one. But minus 70, that's fine. Use that, right? Just keep it simple. Right? We'll do minus 70. We will also see when we get into muscle tissue, we'll see some different things, right? Because muscle is also an electrical tissue. So we're going to look at muscle tissue. We know muscle tissue is different, right, from anatomy. It has a completely different structure, right? But it's definitely acted upon by, right, nervous system, and we use electrical stuff to kind of get a muscle contraction to happen. We'll get there when we get there, right? We're weeks away from that. But we'll see that this idea of membrane potential and resting membrane potential, well, it's also going to factor into muscle tissue, right? So membrane potential everywhere. Resting membrane potential, for now we're focusing on what is that in neurons, right? And we're looking at minus 70 for our most common value. When we get to muscle tissue, we'll see. We'll see some additional, right? What is it for muscle? We'll get there.
So I was just mixing up cellular and tissue. Or cellular and
yeah. So this is the thing, we want to be sure, it's good to have this conversation, right? Because we want to be sure that all the things are in the correct boxes, right? In the head. Then you can study clearly, right? If all the things are in the right boxes.
So I'm piggybacking here.
Yeah.
So Stephen, what is causing that hyperpolarization if it's not, we're not talking about sluggish potassium?
Right. Okay. So let's review. This is a good time to review what is a graded potential. Like, how did we create any graded potential, right? Whether it was the EPSP or the IPSP. What? Actually, let's go back to the picture. Let's go back to our super original picture, which was here. Let's do this. All right. So remember this picture. This is where we started on Tuesday, right? Talking about graded potentials. And how do we create a graded potential? We had our membrane, right? And we already had set up our membrane potential with the sodium potassium ATPase pump, so they just didn't put it in the picture. And we picked a particular ion channel, right? That was ligand gated. And there's our example of a ligand, right? The triangle in this picture. In our picture that we're seeing sort of currently with synapses, it happens to be a sphere, right? But here we saw it as a triangle. But it's the same, it's a ligand. Now maybe we can say it's a neurotransmitter, right? We can sort of make that leap. All right, so we've got neurotransmitter binding to this particular ion channel. So that makes it, first of all, a ligand-gated ion channel, right? So that's our first thing. We have not narrowed it down yet to sodium, potassium, anything, right? Calcium, none of those things yet. We just said ligand-gated. All right, but that's our first incredibly significant difference. That is very significant, right? Because what is the type of gating that we use to create an action potential? Action, voltage, right? Does everybody see that, right? That's huge. If you can see that difference, like the difference between picking a ligand-gated channel and a voltage-gated channel, that's your first, like, separation. That's your first set of boxes right there, right?
So neurotransmitter is voltage gated, or can it be ligand?
Neurotransmitter is ligand,
Okay.
right? Ligand-gated, because neurotransmitter is a chemical, right? We'll see the rest of today what are neurotransmitters, right? But they're going to be all molecules, right? Something, some chemical molecule thing, right? Whereas voltage is all about charge, right? It's electricity. So you see the difference there? Okay, good.
Neurotransmitters are always going to be ligand gated.
Neurotransmitters always be ligand gated. I would say, I think we can say
For
that.
now.
Yeah, I think we can make that statement. Yeah, yeah. All right. So we've chosen to create a graded potential. We've chosen a ligand-gated ion channel so far, right? And our ligand is going to be some neurotransmitter. All right. So far, so good. When that neurotransmitter binds, we get our shape change, right? Here we're opening up, right? Going from closed to open. And whatever ion this particular channel happens to be built for, remember we haven't picked one yet, right? We just picked ligand gated, but we didn't pick sodium, potassium, chloride, calcium. We didn't pick anything yet. All right, so whatever this channel is built for, it will allow that item in. If that item is a positively charged item, like sodium, like potassium, like calcium, right? Any of those things. Then you have positive charge entering in. And if you have positive charge entering in and starting to cling, right, to that inside surface, which direction? Do we go this way or do we go this way? Right, we go this way, right? We go more positive. If that item entering in, let's say it's chloride, which is negative, right? We'd have to have a different setup in the first place, right? Because we've got so much negative charge, but let's just pretend, right? Just to say it. If we had this all flip-flopped and a negative charge entered in, what would we have? Right. The other thing that we could do, again, we'd have to have a different setup at the beginning, but let's just say it, just to say it. Another way to make something go more negative on the inside would be to let positive charge exit, right? Does that make sense? All right. Like I said, we would have to have a different initial setup, right, to get that movement to happen. But it exists, right? We could say, if that were to happen, you end up with that. Good. So do you see how the graded potentials, which the graded potentials our only choices are this and this, right? EPSP and IPSP. You could have big ones, you could have little, right? You could have little or big, but those are the only shapes that we can have. The hills and the valleys for graded potentials. So this is, you know, see how we're kind of limiting our choices, right? You always have to pick ligand gated, and it's going to be some ion. If it's positive rushing in, you get this. If it's negative rushing in, this. So far,
Oh,
good.
so the ISP or IPSP could be a negative charge coming in,
Oh yeah, right.
or
Exactly.
positive leaving.
Right, right. Anything that's going to make this inside surface more negative. Yes, exactly. All right, so that's our graded potential. As opposed to our completely different, separate type of potential, which is action, right? Action potential. We only have one possible shape, right, for action potential, which is this shape, right? Where we have the big mountain peak, and then we have that whoops, the dip at the end of the mountain peak, and then we come back to our starting point, right? Action potential. And to create an action potential, the only way we can create an action potential, only way in a neuron to create an action potential is to pick these two types of ion channels and pair them together, right? They work in tandem. Good? So you have to pick. These are both voltage gated, right? So remember that was in our separate box, right? And one is sodium, one is for potassium, right? And you've got to pair them together so that they can work, right? One after the other. And here is where we see that sluggish potassium channel, right? It is slower in its response to the change in voltage. Good? Good. That's all right. That's all right. No, if there's a dead horse to beat, this is the one. This is the one because seriously, the rest of the course, if you can get this, you can get the rest of the course.
So, okay, I get that part. So
Good.
then figure 6.31,
What-
you're like
what
flipping
side?
from a graded to an action,
Oh, yeah,
and then
yeah.
it's back to a graded potential.
Let's see. Let's pull that one up. So this, yeah, this one. Okay.
So you're switching from a graded potential, then it goes to an action potential because it hits the threshold, and then it comes back down to a graded potential. Is that what we're saying?
Okay. So so we're just focusing on here, right? Okay. So what we've done here, we're measuring. Remember, we're measuring. Here's our little electrode, right? And you measured just that inside surface of the membrane, and they're specifically measuring here at the axon hillock, right? At the axon hillock, because that's where we're going to do all of our adding, all of our summation. So here in this situation, we did boom boom with the A and boom boom with the B, and it all ended up, and boom, we got our action potential. We hit minus 55, and that was enough to trigger those voltage-gated sodium channels to open up, a whole lot of them, and then those sluggish voltage-gated potassium channels could also kick in, and then we have our repolarization and then the hyperpolarization, and then we come back to normal with sodium potassium ATPase pump. So here, you see, this is like a totally like, now let's start over. This is like new situation, right? And so we have like this line, and here's situation number 5, right? So So starting over, right? And then C has some sort of ion channel that would allow more negative charge, right, to build up on that inside membrane, and we have our inhibitory, right, our valley, and that's good, right? That's this,
But this isn't anything to do with the IPSP.
No, this is an IPSP.
Oh, okay.
Yeah, our definition of an IPSP is a graded potential that is a hyperpolarization, that is a downward blip, right? More negative. Good, good. So this is an IPSP. Good. So here we have separate situation, right? Separate situation. Let's let C release some neurotransmitter that's going to result in an IPSP. IPSP. Good. There it is. And then after that, let's try something else, right? Let's let C do that same thing. And you know what? Let's let A release its neurotransmitter. We already know that A is going to give us this EPSP, right? So if we do those at the same time, it turns out that in this example, they happen to cancel each other out, right? Let's say it's positive 3 and minus 3, right? So 3 plus negative 3 is 0. So that's what we're seeing here. We're seeing no change, right? It just stays the same. Good? Got it. All right. Very good. Very good. Very good. Believe me, this is very good. You have no idea. This is very good stuff, and it's completely crucial to have all that stuff we just reviewed extremely clear in the correct boxes, right? All right. So here we're just looking at kind of reinforcing that that those EPSPs, right, and IPSPs are all going to be summed at our axon hillock, right? I actually think I might like this. I might like to redo this drawing as well, and maybe emphasize that charge of the graded potentials, whether they're positive or negative, is really traveling along the membrane, right? I think I would prefer an emphasis on that as opposed to like these sort of swooping lines that are kind of passing right through like the middle of the cell body and kind of passing by like nucleus and Golgi body, whatever, right? Like, I don't know, I don't think I like that. So I don't know, I might go in and redo and make it so that the charges like kind of travel. But the idea of this slide is just to emphasize we're going to add stuff up at the axon hillock. That's what I want you to get out of this. Good? All right. So, oops. There we go. So here we have 2 videos that, I mean, there are a zillion videos out there, but I happen to like these 2 particular ones. They have animations. We're not going to look at them in class just because of time, but you can definitely look at these at home. It's all the stuff that we've talked about. Graded potentials, the E and the I, PSPs, action potentials, a little bit of general electricity information. And but the nice thing is that they're animations, so you know they'll show like a little ball of charge, like moving through an ion channel. It's kind of, I think that reinforces all this stuff that we talked about. So definitely look at these over the weekend as you prepare for Tuesday's exam. I'm not going to pull something out of the videos. You know what I mean, like directly out of the videos, but the information is, you know, it's all complementary to the stuff that we've looked at. All right, so let's talk very briefly about ways that you could possibly control what happens at the synapse, which we know sort of basically is, well, we trigger neurotransmitter release, and we get that neurotransmitter to bind on these ligand-gated ion channels on the postsynaptic membrane. So, starting here at this point and for the rest of today's lecture, we're going to start referring to these ligand-gated postsynaptic ion channels, right? that we already know are in charge of creating graded potentials, either EPSPs or IPSPs, right? We know that. We're going to start referring to them as receptors. We're just going to start using that word instead of saying ligand-gated, synaptic, what else did I say? Ion channel, right? That's pretty long. So we're going to start to say receptor. You could say postsynaptic, but really in practice, everybody always just says receptor, right? Receptor. So we're going to be learning, right, for the rest of today, some specific neurotransmitters and their specific receptors. Good. All right, so that's our new vocabulary word for today. So how can we control, this neurotransmitter release and this binding with receptors to create our graded potentials over here? I don't know, we can do a bunch of things. One big thing is that we can take a completely separate axon terminal and actually allow it to have its own synapse with this main axon terminal, right? This is like the one that we're focusing on, our biggie. And we can take another axon terminal and create like little mini synapse, little side synapse, right? And this little side synapse or mini synapse will control what's going to happen at the big main synapse. We call this, like I put up over there, we call this an axo-axonic synapse, like axon to axon. That makes sense. All right. But it's going to act kind of the same way, like the mechanism of it will be kind of the same. We'll have an action potential arrive, that will trigger, they're not showing it here, but we know that we would open up some voltage-gated calcium channels. The calcium would trigger these synaptic vesicles to release this neurotransmitter onto these receptors, and then these receptors would create some kind of graded potential that would travel over to here, do something in here to trigger this release, and then boom, we can keep going, right? But this will influence the main action happening here. So that's one way of controlling what happens at a synapse. Let's see, another way we can control what happens at a synapse is to insert receptors on the presynaptic membrane, so on the axon terminal itself. We're going to call these autoreceptors, like self-receptors. And you see how these autoreceptors have the same shape of binding site as the regular postsynaptic receptors, right? We have the same shape. So we could have neurotransmitter bind onto our postsynaptic receptors, get our graded potentials. All right, great. We've done our job, right? And then maybe this neurotransmitter is going to be released, right, back into the synaptic cleft, and maybe it'll come back over here into these autoreceptors and trigger this axon terminal to do something. Maybe let's take back that neurotransmitter, right? Maybe trigger some sort of endocytosis, right? And re-uptake that neurotransmitter. That would be kind of good because then you could kind of clean up all that neurotransmitter, right? Not leave it hanging around. Good. You could do that. That's like an example. All right, so that's something we could do. Let's see. I think on our next slide, yeah. On our next slide, we just have even more ways that we could control what happens at a synapse. And here we're going to add to our repertoire not only stuff that's built into the body, but we're going to say, oh, we could maybe either take or administer some drugs. Some of them could be like legit, some of them maybe not, right? Or we could maybe even have a disease process, like an example would be some sort of bacterium releasing maybe a toxin or something and affecting things in some of these steps. So maybe here's an example of a step here. They're telling us this is a synthesizing enzyme, an enzyme that would synthesize maybe a neurotransmitter, right? So if something, either a drug or a toxin or something, would somehow work its way to here and maybe block this enzyme? Maybe turn it off or slow it down or something like that. Well, if you can't make your neurotransmitter, you can't release it either, right? And then, whoops, that's not going to happen, right? All this stuff with the synapse. So you see how that's, right? That's something you can do. So, so you can kind of look through, right? Just all these different locations or all these different locations that we already know where a drug could act or a toxin, right, from a bacterium could act, right? So you can read through all of those. What I would like to do is look at two specific examples of two toxins that come from two types of bacteria. On our exam, so this exam shows up Tuesday, right? This exam shows up this Tuesday. On the exam this Tuesday, I will not test you on, this is micro. This is microbiology right here. But I include it, because that's for micro, right? But I include it here because in certain situations in physiology, sometimes looking at what goes wrong can help you to better understand what goes right, right? How do things work when things are going right, right? So this is one of those cases. So I do want to take a look at it, but I won't test you on it, because that's really not. But let's see, we've got two bacteria. They are related to each other, but they are different. This one is the bacterium that will result in a disease called botulism. And here we've got a bacterium that will result in a disease called tetanus, or a situation called tetanus. And here are the two toxins that each one produces. They're different toxins. We've got botulinum and we've got tetanus toxins. And these are neurotoxins, meaning they like to go to, they like to target, right? They like to work at neurons, right? Neurotoxins. So that totally makes sense. Once they enter into a neuron, they both are actually going to work in pretty much the same way. They're both going to attack, remember those snare proteins that were the proteins that would help us with exocytosis, right? And we need exocytosis at the synapses to release neurotransmitters, right? So if we're going to attack those snare proteins, we're not going to be able to release neurotransmitters, right? No exocytosis, no release of neurotransmitter. So we've got two different toxins produced by two different bacteria, right? Though they're related, the bacteria are related. And these two toxins, once they enter into the neurons, they do work in the same way, right? The same sort of subcellular mechanism of destroying the snare proteins. But these two different neurotoxins act in that way in two different locations in the nervous system. So this is how and why we see two different things, right? Two different things happening in the body. So let's take a look at that. Here, for tetanus, this particular bacterium is going to, when it enters in, it's going to travel to central nervous system neurons, right, so far so good, that are the ones that normally, when they're working, they inhibit the neurons that are the ones that activate skeletal muscles. So let's try to piece that together with the anatomy, right, that we are going to try and remember right now. Remember we had, of course, skeletal muscles, and we were going to have motor neurons controlling those skeletal muscles. And motor neurons are always going to tell skeletal muscles, go, contract, right? They always say contract. And we should remember that piece of information for physiology, for sure. Because we're going to talk about muscle tissue next anyway, right? So let's just, we'll keep that in our heads. All right, so motor neurons tell skeletal muscles contract. Always. That's the only thing they can possibly tell a skeletal muscle. Okay, if a motor neuron can only tell a skeletal muscle contract, how are you ever going to relax that thing? You have to turn off, right, or inhibit that motor neuron, right? And then you can relax, right? If I keep pointing this bicep, Good? Okay, so far so good. So, the tetanus bacterium targets those inhibiting neurons, the ones that tell the motor neurons, shh, right? Turn off. Don't do anything, right? Don't tell the muscle to contract, right? So we can relax. So if we've got a neurotoxin attacking the snare proteins in those inhibiting, right, controlling neurons, right? What is it that is not going to be able to happen? If we can't tell our motor neurons, shh, what can we not do? We can't relax, right? So now we've got this bicep muscle, right, that no matter what I do, right, no matter what I think, oh, can't relax it, right? We're going to give that a name. We'll call it spastic paralysis. So spastic or spasticity is our official word for not being able to release a contraction, right? The contraction is stuck. And paralysis is our word, like most people know, right? Paralysis is when you can't move, right? So, but most people think of paralysis like, let's say if someone's in a wheelchair, right, and maybe they're paraplegic and they can't move their legs, right? Most people think of paralysis like that. But this type of paralysis results from, oh my gosh, a muscle that can't relax. If you can't relax your muscle, you still can't move, right? If this is completely stuck, I still can't move my arm, right? All right, so that's this word, spastic paralysis. All right, keep that in your head for a second. Let's compare that to the botulism, right? The botulinum bacterium. So this bacteria is going to target, the neurons that actually activate the skeletal muscle, the motor neurons, right, that are telling the muscle to contract. So if we've got a toxin destroying the snare proteins, right, of those neurons, what can we not do? You can't contract, right? So now we're stuck here. No matter what I think, right? Nothing is happening. So we're going to refer to that. It's also paralysis because I can't move, right? But we'll call it flaccid, which is floppy, right? You're floppy. So floppy, loose, right? Paralysis. All right, so now we've got these two different types of paralysis. They're like opposites, right? They're both paralysis. We can't move. We're stuck. But one is because the muscle can't relax, and one is because the muscle can't contract. So two opposite things. If we ended up with either of these types of paralysis in the diaphragm, oops, exactly, no good, incompatible with life, right? If the diaphragm contracts and we can't relax it, we needed that relaxation to take our breath in. Whoops, all right? And then if we can't contract the diaphragm, whoops, right? You see it? All right, so either way, ultimately you end up with the same result, right? No breathing. Anyway, this is our example, right? This is our example. But like I said, I'm not going to test you on the specifics of that, but I hope that that example helps us to understand, right, some stuff about like what's normal, right? How does stuff work in a normal situation? I hope. All right. So here we just got a summary of different types of things that could affect action at a synapse or how strong the result is at a synapse. Some of this stuff we mentioned, like your axonic, right, synapse, your autoreceptors. Some of the stuff is stuff that we won't get to maybe for a while. There's a down regulation thing again, right? So I don't think I would actually worry too much about this table now that I look at it. Yeah, it's just they mentioned some of the things. I might even remove this table. I don't really like it here anymore. Now that I think about it, let's forget it. I actually don't like that table. Okay, let's move on. Oh, okay, but we're gonna love this table. It's a dub table. It's a dub table. Okay, let's just look at it and then we're gonna take a break, right? And then after our break, we'll do all the like the dub table. All right, so but just to say it before our break, this is our table with neurotransmitters. Remember, it's like I've been kind of setting you guys up for like, oh, today we're gonna talk about neurotransmitters, right? So, this is a table of neurotransmitters, right? And we're gonna know this table. And we're gonna know details about the items listed on the table. So at the moment, this table is an intro to our whole neurotransmitter section, which is the rest of today. We will see this table over and over and over again for the rest of today. By the time we get to the end of today, which would be the bottom of the table, we will be able to look back at the table and it will be a review, right? So this is a great way to test yourself as you prepare for Tuesday, right? You just look and you say, norepinephrine, what do I know about norepinephrine, right? That's what we're going to talk about after the break and all these other things, right? And you can, what do I know about this glutamate, right? And you can think to yourself, right? Let me think, what do I remember? Good? So this is also going to be your, like, testing yourself review table. Good? All right, so let's take our break. Let's go till 9:25, and then we'll come back and get started on all these neurotransmitters. It's tough stuff. It's tough stuff. Wow.